Hi everyone. Today I will present our work entitled Comparative Survey of Multigraph Integration Methods for Holistic Brain Connectivity Mapping. This work has been done at Basira Lab by myself and supervised by Dr. Hatice Camgöz Akdağ and Dr. Islem Rekik. The interaction and relationship between different cortex regions in the human brain can be measured using different types of connectivity. Functional connectivity derived from functional magnetic resonance imaging fMRI, structural connectivity derived from diffusion weighted imaging DWI, and morphological connectivity derived from structural T1 weighted MRI imaging. Using different sources of measurements, one can derive for the same subject multi-view brain connectivity networks where each view corresponds to an imaging modality or measurement quantifying a single type of brain connectivity network. Analyzing such multi-view connectomic dataset together remains challenging due to the large intermodality variations across different views of connectivity networks and their heterogeneity across the population subject. Nonetheless, mapping brain networks of a whole populations into a single representation named Connectional Brain Template or CBT is substantial for capturing the most representative and shared brain connectivity patterns within a heterogeneous network population, acting as a connectional fingerprint. Furthermore, the state-of-the-art have shown that by comparing the CBTs of two populations, we can easily identify a reliable biomarkers or the most discriminative regions that disentangle the difference between two populations. Example, disentangling the typical from atypical variations across healthy population and disordered population individual. Brain of an individual can be represented by single view or multiple view brain network, each representing a specific feature derived from different neuroimaging measurement. Given a population of single or multiple view brain network, we are aiming to review the existing integration methods that generate brain fingerprint of the population, which can be represented by an integral connectional brain templates named CBT. We are also aiming to compare the method's CBT performance. In the case where individuals in the population are represented by a single view brain network, we review three integration methods. Similarity network fusion or SNF is a nonlinear diffusive shrinking technique to integrate single view brain network population. Network Atlas Guided Feature Selection or NAGFS is a feature selection technique to diffuse and fuse single view brain network population. And supervised multi-topology network cross diffusion or SMNet fusion propose kernel clustering methods followed by a nonlinear fusion using weighted multi-topological measures. In the case where individuals in the population are represented by multi-view brain network, we review 5 integration methods. SNF clustering average or SCA use nonlinear fusion followed by clustering technique. Network normalization or NetNorm employs feature selection technique prior to a nonlinear fusion. Multi-view clustering and fusion network or MVCFNet introduces clustering methods based on manifold learning followed by linear and nonlinear fusion. Clustering-based multigraph integrator network or CMGINet employs multi-kernel manifold learning for clustering and deep learning graph embedding for multi-view brain network integration. And Deep Graph Normalizer or DGN utilizes geometric deep learning to normalize and integrate multi-view brain networks in an end-to-end fashion. Our work is an in-depth survey where we review the state-of-the-art integration models for single-view and multi-view brain network, and a primer comparative study that evaluates their generated CBT's learning performance in terms of well-centeredness by achieving the minimal distance to all individuals in the population, discriminativeness by capturing distinctive population connectivity biomarkers, and topological soundness by preserving the topological properties of the population individuals at different scales, including node-wise similarity, distance similarity, and global similarity. We ran an extensive experiment to compare different facets of all the reviewed methods using complementary evaluation metrics to make sure that our conclusions are robust. To evaluate the learning performance of CBTs generated by the integration methods, we include the following measurement. First, we looked at the CBT centeredness using Frobenius distance. Additionally, we examined the CBT discriminativeness using the overlap between the top discriminative arrows, as well as the CBT topological soundness in terms of distance-based level using Hamming and Jacquard distances, global-based level using global efficiency and modularity, and node-wise using local efficiency, participation coefficient, and 8 centrality metrics with their KL divergence. Our extensive comparative study includes three-way evaluation strategies to assess various aspects of the brain connectional template while comparing the different integrations methods. The first is methods to methods analysis, the second is methods based analysis, and the third is arrowwise based analysis. Combined together, these strategies enable the investigation of centrality, discriminability, and the topological strength preserved by the generated CBTs. We conduct our comparative study between different CBT integration methods using two datasets. The Genomic Superstruct Project dataset is composed of 308 female subjects and 391 male subjects. The Alzheimer's Disease Neuroimaging Initiative dataset counts 70 subjects with Alzheimer's disease and 64 subjects with late mild cognitive impairment. For both datasets, each subject is represented by 4 cortical morphological brain networks derived from mean cortical thickness, mean sulcal depth, average curvature measurement, and maximum principal curvature. For both hemispheres, the cortical surface is reconstructed from T1 weighted MRI and parcellated into 35 cortical regions of interest or arrowwise for left and right hemispheres. To assess the reproducibility of the CBT produced by each integration method, we utilized 5 cross-validation. To ensure the robustness of our conclusions, we employ a total of 18 evaluation measures throughout our experiment. In order to avoid a lengthy presentation and overcrowding, we only present the results of the experiment using the Brain Genomic Superstruct Project. The result of the experiment with the Alzheimer's Disease Neuroimaging Initiative ADNI dataset can be found in the supplementary materials. Here, to test CBT centeredness, we report the mean Frobenius distance for each cross-validation fold as well as the average across folds between the connectional template generated from integrations models. For single-view fusion methods comparison, SMNet fusion significantly achieved the lowest mean Frobenius distance to the population's single-view networks. As for the multigraph integration methods comparison, DGN significantly achieved the lowest mean Frobenius distance to the population multi-view network, showing that the generated CBT is more representative and well-centered. Here we test the CBT's discriminativeness by computing the matching rate in percentage between the top 15 and top 25 most discriminative arrows identified using interclass CBT difference and those using the combined super-vector machine multi-kernel learning classifier. The results demonstrate that for single-view brain network, SMNet fusion significantly reached the highest overlap percentage with respect to other single-view integration methods, while for multi-view brain network, DGN significantly generate a more well-discriminative CBT than other multigraph fusion methods, able to easily spot both gender-distinctive brain regions. We test the topological soundness of the generated CBTs using topological measures across regions of each CBT against the ground truth calculated as the average across views and testing samples of a given measurement. On the global level, we use the modularity, which detects the goodness of clustering nodes into communities where dense connections between nodes are within the same clusters and sparse connections are in different clusters. For single-view fusion comparisons, SMNet fusion outperforms other single-view integration methods. While for multigraph brain network, DGN significantly achieved the highest score. On the same global level, we use global efficiency metric which quantify the exchange of information across the entire graph. Likewise, for single-view fusion comparison, SMNet fusion outperforms other single-view integration methods. While for multigraph brain network, DGN significantly achieves the highest score. On the node-wise level, we employ participation coefficient to evaluate the integration behaviors of the ROIs in the brain network. Participation coefficients quantifies the distribution of the node edges among the communities of a graph. Likewise, for a single-view fusion comparison, SMNet fusion outperforms other single-view integration methods. While for multigraph brain network, DGN significantly achieves the highest score. On the same node-wise level, we use the local efficiency measures to evaluate the segregation behavior of the ROIs in the brain network. The local efficiency quantifies the efficiency of information transfer limited to neighboring nodes in the graph. Likewise, for single fusion comparison, SMNet fusion outperforms other single-view integration methods. While for multigraph brain network, DGN significantly achieves the highest score. In summary, both DGN and SMNet fusion integration methods exhibited superior performance compared to other methods in terms of global and node-wise metrics, achieving the highest score for modularity, global efficiency, local efficiency, and participation coefficient. This can be attributed to their ability to maintain connection strength across the entire brain graph structure for all subjects, thereby preserving the brain graph community. Additionally, these methods maintain information transfer between neighboring nodes while fusing single-view and multigraph networks from the population. To assess the happiness behavior of the ROIs in the brain network, we perform additional evaluations of the topological soundness. This involves comparing the similarity of the generated CBTs with the ground truth using 8 centrality metrics: betweenness centrality, eigenvector centrality, information centrality, Laplacian centrality, node strength, random walk, cat centrality, and page rank. Betweenness centrality measures the node importance considering its connection with direct neighbor and detects node with the shortest path between all pairs of the nodes in the graph. In the case of single-view fusion, all methods fail to preserve the single-view connectomic data topology. While for the multigraph integration, the connectional brain template generated by DGN show a striking similarity with the ground truth data in the topological properties, showing DGN ability to capture more complex topological patterns. We also compute the eigenvector centrality to quantify the indirect node strength connections of the CBTs. Again, none of the single-view methods show any similarity to the ground truth. However, the CBT generated by DGN shows a strong happiness behavior with the ground truth data in the case of multigraph integration. We also employ the cat centrality to measure the CBT node's importance while considering their direct and indirect neighbors. Similarly, among all integrations methods, only the CBT produced by DGN was able to preserve the topological properties of the multi-view brain network population. In addition, we quantified the impact of node removal from the graph to investigate both local and global node importance. Likewise, only the DGN method succeeded in preserving the importance of both local and global nodes while integrating the population of multigraph brain network. Furthermore, our experimental results indicate that by computing the information centrality metrics, the CBT generated by the DGN methods preserve the closeness of its nodes, in terms of resistance and reachability to the others, unlike other multi-view fusion methods. Our experiments further showed that the CBT produced by the DGN methods maintains the count and the strength of edges linked to each node, as revealed in the PageRank matrix. Moreover, we found that the DGN methods preserve the weights of edges connecting a node directly to its neighboring nodes, as indicated by the CBT's node strength matrix. We extend our evaluation to investigate the dissimilarity between each of the distributions of centrality metrics derived from the CBTs and the ground truth distribution using KL divergence, where small divergence signifies similar distribution. Our experimental results show that for single-view integration methods comparisons, SMNet fusion achieved the lowest divergence score on gender dataset and the Alzheimer's disease neuroimaging initiative using the left and the right hemisphere and across all topological measures. While DGN outperformed other multigraph fusion methods as demonstrated by achieving the lowest average KL divergence distribution on both datasets for the left and the right hemisphere and across all topological metrics. We also test the topological soundness of the generated CBTs on the distance level by computing the Hamming distance between the CBTs to evaluate the amount of changes between two of them by counting the number of edges, dilation, and insertions necessary to fully transform one CBT into another. Remarkably, the CBT learned by the DGN methods stands out with the highest Hamming distance among CBTs generated by multigraph fusion methods. In our comparison of single-view integration methods, we did not observe any difference in the generated CBT. We further evaluate the distance level of the topological soundness of the generated CBTs by comparing their Jacquard distance to quantify the proportion of edges that have been removed or added with respect to the total number of edges appearing in either CBTs. Likewise, and notably, the CBT produced by the DGN methods exhibit the highest Jacquard distance compared to the other CBT generated by other multigraph fusion methods. We can attribute the reason of DGN generating a more dissimilar CBT than other integration methods to its normalization of multigraph network in an end-to-end manner using geometric deep learning. For the single-view integration methods comparison, we did not observe any differences in the generated CBT. By comparing the CBTs generated from the DGN methods for populations with Alzheimer's disease and late mild cognitive impairment in each hemisphere, we can easily identify the top 3 biomarker regions that distinguish between these two brain states. Both left and right hemispheres share the same biomarkers. First, we distinguish the entorhinal cortex responsible for the function of the memory. This region has decreased volume and thinner cortex in AD rather in LMCI. The second region is cuneus cortex, which has a thinner cortex thickness in the AD rather in LMCI. The third most discriminative region is the temporal pole, where previous studies have shown a cerebral atrophy through the evolution from LMCI to AD. By comparing the CBTs generated from the DGN methods for female and male populations in each hemisphere, we can easily spot out the top 3 biomarker regions that distinguish between gender difference. For the right hemisphere, we distinguish the superior parietal cortex region, which is responsible for the visuospatial task performance. Previous studies show that this region has a higher activation in male rather than female. The second most discriminative region is corpus callosum, where it has been shown that this region has a bulbous shape in women and a more tubular shape in men. The third region is inferior parietal cortex, where it has been proven that female has a thicker cortex in this region rather than male, with larger volume cortex. For the right hemisphere, we distinguish the rostral middle frontal responsible for analyzing visual reasoning tasks. Previous studies show that in this region, male has automatic process while female has top-down process. The second region is lateral occipital cortex, responsible for face expression. Study prove that female has higher activation in terms of rapid visual time rather than male. The third discriminative region is precentral gyrus, where male has shown an increase in learning disability comparing to female. In summary, and in this work, we introduce the first comparative study of integration methods for single-view and multi-view brain networks to estimate a connectome population fingerprint. To evaluate the performance of the integration methods, we conducted extensive experiments in terms of centeredness, discriminativeness, and topological soundness, using similarity measures at the node-wise level, local and global levels. In our future work, we will evaluate a time-dependent multigraph population, include non-isomorphic graph, and integrate multimodal brain networks. Feel free to check our review paper on comparative survey of single-view and multigraph integration. You can also visit the GitHub repo of the paper to generate similar plots. For more details, please visit us at basiralab.com website and YouTube channel. Thank you for your attention.